EEG kind of given some warnings about the versions because most of it's been a while that I've already downloaded this EEG. So I'd like to be updated with the newest version of this software. I recommend that you use EEG Lab's official website to download the newer version. But there are a lot of plugins that you may or may not have in the default version. You may have to go to, let me show you. You have to, I don't know if maybe this is the best way to start the conversation. So if you go to manage EEG Lab extensions, what's happening to the screen?
It's going to die in some days. Feel like that because we are getting the stripes there. And
oh, I see. Okay.
Last.
Oh,
Last
breath. Oh my goodness. Okay, gotcha. Okay. Anyway, we were just discussing the display. The display we're using in the lab, showing some signs of sickness because it gets dark, and then I can't see the screen. So, but anyway, what I was referring to a minute ago, that if you go to these extensions tab, you will realize that there are a lot of plugins that you are able to download and set up within the EEG Lab framework. And some of them are already there. And if you're unable to find some of them, like for example, IC label that I already downloaded and set it up, that we will be using for the automation of labeling the independent component analysis after the ICA. We will have a lot of components, and I'm going to be talking about them today. Then we will label them, whether we would like to include them in our analysis or exclude them from the analysis. But the IC label is a tool that allows you to do this labeling automatically, like, based on certain set of thresholds. But anyway, I don't have time to go over all of them, but hopefully I will touch upon each one of them as I proceed in my presentation. But please interrupt me if you have any questions. And today I'm hoping to get our hands dirty with the EEG Lab software. So we will be processing a real EEG signal today together. So I don't want it to be purely theoretical or just PowerPoint slides. Okay, so let me go back to my presentation. And I'm hoping that everybody is able to see my screen. Is that correct?
We can see it, so
they should. Okay.
Yes.
All right. So let me first. So this is the second episode of our EEG analysis series. So therefore, I'm already covered maybe a large chunk of pre-processing steps on the multivariate time series data, such as EEG. And I'm going to first give you a brief background on the challenges of EEG for those of you who come to the second session directly without watching the first episode. So first and foremost, EEG analysis is quite challenging because there are pervasive artifacts, and particularly, as you might know from our previous episode, that EEGs can be collected using, without invasive approaches. That means there might be a lot of noise in the EEG signal, and therefore that would cause a lot of pervasive artifacts that we need to deal with. And EEG recordings are typically mixtures of all brain activities arising from different networks. In other words, some of these components of EEG are related to the task that you're trying to measure. Some of them are not related to these tasks. So therefore, you need to be cautious about the EEG signal you collect on your experiment and be able to identify which of these components are relevant to your task. And response variability is another challenge. Unfortunately, response could be varying across trials, and it can actually vary across subjects. So there is this variability issue that you need to be aware of. And another challenge that I'm going to be emphasizing today is the inverse problem. So let me just summarize what it is, but the ensuing slides in my presentation will cover more details about the inverse problem. Inverse problem is in a nutshell is about estimation of the source signals which are in the stem in the brain. In other words, they're not necessarily in the cortex. So we would like to, based on the EEG signals that we collect across different electrodes that's spread across the scalp, we would like to estimate the source signals, meaning that the neuron populations in the stem that generates these electrical fields, and then that causes the expansion or maybe propagation of electrical field that can be measured on the cortex, in other words, on scalp. You using either gel to create a conductive channel between the scalp and the electrodes, or you can use dry electrodes, directly capture the signals on the scalp. But just so you know that these electrical signals are not necessarily originated from the cortex, they may even be originated deeper in the brain, and we would like to estimate those sources to be able to get reliable information of where exactly in the brain the neuron populations are activated based on what task, right? The problem, when we call inverse problem, is that since this estimation is in inverse direction, so we don't have access to what's happening inside the brain, we're trying to estimate those sources. That's where the inverse comes from. Because as you shall see in my next few slides, that the forward problem is, the source signals, you basically have a mixing matrix that will multiply these source signals to generate the EEG signals. This could be useful if you, for instance, simulate a brain to generate these EEG signals, and then compare the real EEG and the simulated EEG. See if your simulator works well. So that's the forward problem, how we're going to mix, for instance, these source signals to get the EEG signals that are compatible or congruent with the real EEG you collect from your experiments. The inverse problems, on the other hand, is computation of these, estimation of these sources based on the available EEG data that you collect from your experiments. And there are other, maybe a vast list of other problems that are associated with the EEG, this specific modality. But I'm going to jump because I've already started covering most of the basic pre-processing steps so far, and I'm left with few others. So I'm going to basically go over all these extra pre-processing steps that are necessary to complete the standard pre-processing phase. So I'm going to go through them, and I'm going to probably rush through them. So if you have any questions, please, or something that you want to maybe explore more, just let me know so I can give more information. So I'm not going to necessarily going to provide all the information, because these for example, source localization, it can be taught in an hour class, with examples and etc. I've got also a lot of slides, but I'm going to probably skip them just so that we have more room and space and time for practical examples with EEG lab. So in the source localization problem, as I said, there are two types of problems: inverse and forward problem. The solutions, for example, for the inverse problem, how about the solutions are very challenging because you think about a simple linear equation like x equals a s. A is matrix, s is a vector, and x is also a vector. So in the inverse problem, what you see is basically x, which is the EEG signal that you collect from your experiments. And question is, what is A, the matrix, the mixing matrix? Because if you calculate mixed matrix, then you can actually multiply the inverse of that matrix, the EEG signals, which is X, to estimate the source signals, right? So the challenge here is that you don't know what A is, the mixed matrix, as well as the source signal. So you don't know A and S. Like if you've taken like elementary or my fundamental linear algebra class, you would know that if you know A and if you know X, it's trivial to calculate S as long as A is full rank, right? However, in our problem, this real world problem, we have no access to A and neither to S. So therefore, we need to make assumptions about the mixing model. That actually corresponds to choosing a model. For instance, you have to make assumptions about the spherical shape of the sculpt, how many dipoles you have, dipoles meaning the source that generates these EEG signals, or what is the physical, like, for example, I talked about propagation of electromagnetic signal through different mediums. Like this medium could be the skull, or it could be the tissue, right? The brain tissue, or the white matter, and the propagation of electromagnetic signals across these different mediums, the speed of which could vary actually based on the physical features of these mediums. So therefore, you have to make assumptions about those physical mediums too. For example, a finite element model or the current density change, across these mediums. These are all subject to making the set of making assumptions. If you use standard methods, the spatial resolution is particularly low. In other words, calculation of the exact location of these sources is pretty challenging, maybe on the order of 2 and 3 centimeters, which is pretty, a bad estimation. That's why, but there are also some fancy methods, which I don't think I can dive into them today, but maybe in a later session, that can expand the spatial resolution that is available in the EEG signal. Because EEG signal, as you might imagine, usually is popular for its time resolution, not spatial resolution. Spatial resolution, for example, if you want to get really good spatial resolution, you usually go with fMRI. But EEG can also provide some spatial information. And if you would like to boost that spatial information higher levels, then you have to employ more fancy methods to get to, for example, millimeter resolution. So the blind source separation problem is actually a large, broad concept. And an independent component analysis is one of particular method that finds underlying factors and components from the multivariate data based on an assumption. And an assumption is the independence of the components. In other words, we would assume that the sources are statistically independent and they are not Gaussian. Okay, so that's the specific assumption that basically separates RCA from all other methods. For example, PCA, maybe some of you guys might be familiar. The PCA is another method that is also a linear method that seeks directions that represents the data best in the variance sense. In other words, if you think about the signals and it's in the variance space, you can actually decompose that signal into different variances that are orthogonal to each other. And you can think of the variances, if you think about the variances as arrays, for instance, with certain direction and magnitude, then the question with the PCA asks is, how can I represent the signal using these orthogonal arrays? Just like we found in the linear algebra, the basis functions, right? So your first variance direction is the most significant one. And then the second one is the least significant, and so on and so forth. And then you find the weights to be able to represent your signal and how much variance you have. PCA is really good, for example, identification of how much noise you have in your time series. If you're, for example, if you are given 2 different time series data, and if you run a PCA on these 2 different signals, and if you find that the first signal has And for example, if you'd like to retain only 90% of variance in the signal, and then you throw away the rest of the 10%, and if you find the first signal is represented by 10 different components, whereas the second is only 3 components, then you can reliably say the first signal has more noise in it because you require more components to represent that signal. So PCA is good at, for instance, measuring maybe weakly the noise contained in your original signal. ICA, on the other hand, thinks directions that are most independent from each other. Okay, so statistical independence is the core distinction of ICA. If somebody talks about ICA, I think they start with talking about cocktail party problem. In this simple problem that you have multiple sources and the observations are mixtures of these sources. You can think of this as in the sound field or in the image field, but these sources generate multidimensional data, not necessarily single-dimensional data. But then there is a certain mixing matrix that mixes your sources and then creates these observations. In our EEG case, for instance, the sources are brain source signals, and then the mixing matrix is dominated or defined by the medium. That's the channel due to brain's biological structure, and finally the observations are the electroencephalography signal, EEG signals, and we model this mixing phenomena or cocktail party problem using a simple algebra, simple linear relationship, whereas the sources are represented by S as a vector, and the matrix is A, and then the X is the observations, which is EEG signals. So ICA, in this particular simple format, does the reverse work. What does that mean? That means we observe X, and which is hypothesized to be a linear multiplication of the source signal and the mixing matrix. And the task is we would like to estimate both A and S. Okay. So for that to happen, we have to find an inverse of A, which is predicated upon the existence of inverse, right? So A cannot be degenerate. So you have to estimate the inverse of A and then multiply with X. X is the observed signal, so therefore it's available already, so that you can estimate S. So the main idea here is to estimate the matrix, the mixing matrix first, and then you estimate the source signal. Okay. On the right, you see an example where I have 2 different independent sources, and they are not Gaussian, as you can see. One of them is like simple sinusoidal signal, the other is square wave. And then I have 2 mixtures with different weights, which are characterized by these symbols A1, 1, A1, 2, A2, 1, and A2, 2. And we would like to find, for instance, the mixing matrix A. As I said before, the problem is ill-posed. In order to approach to that problem, you have to make assumptions, and our assumption in the ICA is to assume that the sources are statistically independent, and also they are non-Gaussian. Okay, so as I said before, ICA is one type that has got this independency constraint, but there are also other types of blind separation algorithms, source separation algorithms. PCA is also one of them, but you may already know what it is, and also I've explained it a bit. But there are also two other forms of constraints that you can impose on your solution. One is called sparsity constraint that leads to SCA, okay, sparse component analysis. And the other is non-negative matrix factorization. That has a non-negativity constraint. So sparsity constraint, or SCA, is more a practical solution, actually preferable solution to ICA, if your number of sensors, in other words, your electrodes in our EEG context, is less than the number of sources. So you can actually come and say, how many sources? I have no idea how many sources I have. Yeah, that's a correct actually, it's a very accurate question, very legitimate question. And but there are two approaches to find the number of sources. One is based on your previous background on the task that can actually elicit these neural populations to elicit physiological signals, or you can run fMRI study to identify how many sources are involved in performing such tasks. But if you've got a background, like a priori knowledge about how many sources you have, then you can choose among these different constraints. Like ICA is good if the number of sources is less than the electrodes. That's why, for instance, in the EEG experiments, we typically want 32 channels, or maybe more than 32, 64 channels, to make sure that the number of sources is less, strictly less, than the number of sensors we have. And in that case, ICA is a better methodology. But if it's the other way around, number of sources are bigger than the number of electrodes, because of your practical constraints of capturing EEG signals, particularly in mobile context, that might be the case. In that case, maybe sparsity constraint could be a better solution, and sparse component analysis could be a better technique. Okay, so the cocktail party problem, also blind separation problem, is a degenerate, as I said. And the most common assumption is that source signals are statistically independent and non-Gaussian. And the methods based on this assumption are called independent component analysis methods. So it's kind of a summary what I said. And these statistical techniques of decomposing a complex data set into independent parts, is probably the most complex part of the pre-processing, as you will see when we do the analysis ourselves in the MATLAB. And it can be shown that under some reasonable conditions, if ICA assumption solves, then the source signals can be recovered up to permutation and scaling. In other words, the ICA generates a unique solution up to permutation and scaling. However, for instance, SCA unfortunately cannot generate a unique solution. And one of the other disadvantages of SCA, for example, is more complex, because you're not solving a convex problem. In sparsity constraint, you use your cost function is basically looking at the absolute difference rather than, for example, squared difference. That basically increases the complexity of the problem and makes it non-convex, unfortunately. So that is part of the reason why the sparse component analysis is a more complex technique compared to ICA. Let me just skip this part. If you're interested in knowing how ICA works, I can actually talk about that. But I'm going to just give you an example of illustration of ICA with two signals. So consider there are two signals, S1 and S2. Hopefully they're independent, otherwise ICA wouldn't work. And also they're also hopefully non-Gaussian. So we have these mixing matrix, right? A1 and A2, A11, A12, A212, and 22. And as a function of time, they change over time. So what we do basically, we mix these signals using this A, mixing matrix that basically changes the whole landscape and its representation of two-dimensional space, as you can see. But now we have these X1 and X2, right? So these are observations now. And we're trying to estimate S1 and S2 from X1 and X2. So as you can see, there is this linear transformation that puts S1 and S2 to X1 and X2. We call them mixed signals. So the first thing that we would like to do is to identification of these two different directions you can think of. There are two different new axes, basically, that defines X1 and X2. And these are characterizing the matrix, mixing matrix. So what we want to do, basically, we would like to find first the rotation. What's the needed rotation to get this A1 and A2 in place such that it's close to the original axis. And this is the step one. And then step two is after rotation. Actually, this is unfortunately a typo. After rotation, you basically do inverse rotation to be able to get the signal to its original axis. So what we're basically trying to do here is trying to estimate the original signal, which is here. And in the first stage, we're trying to do the uncorrelated, because independent signals or statistically independent variables are bound to be uncorrelated. So the first step in this technique is to make it uncorrelated as much as possible, and then apply this rotation to put it in the same axis with the original signal. Okay, because without resolving or making the signal uncorrelated, you cannot ensure that it's independent. And this rotation component makes sure that it's in the same axis. The ICA does not necessarily provide you the true solution, right? It basically attempts to make these S1 and S2, the original source signals, as independent as they can be. Okay, just so you know, because the mixing matrix could be non-invertible. You know, that's a possibility. Particularly, that depends, the invertibility of the matrix A depends on your pre-processing steps. For example, if you don't apply, for instance, any lower high pass filtering, then your signal could have a DC component, and DC component is basically common to all the channels, right, in your EEG, making them correlated with each other. Okay, and therefore source could be correlated by the same logic. Okay, and also non-Gaussianity is also an important prerequisite for this technique to work, because as you can see, if you have a Gaussian signal, then rotation cannot make it uncorrelated. Okay, so therefore Gaussian assumption is not a true assumption, or if your signals are Gaussian, the IC is not the best hope for you. Okay, just so you know. Anyway, there are lots of applications of blind source separation, and we will be focusing on medical signal processing, particularly EEG. But there are other fields of application. For instance, here you can see that people used ICA to denoise the images, and you can see the ICA filtering could actually give you better performance compared to binary filtering. And the reason is that ICA is able to decompose the signal into noise component as well as the source component, source signal component. And it basically removes the noise component to reconstruct the original image. Okay, so here's what we had last time, and we would like to continue applying ICA to remove the artifacts in our signal. Here is an example of the original signal on the very far left, and the components on the right. After you calculate the inverse of the mixing matrix, you multiply. Let me see if I can use my. Oh, okay. Here, I think you can see my cursor. So this plot here is outcome of the mixing matrix, the inverse of the mixing matrix, multiplying your channels here, multiple channels, and then you get this first component, second component. The components are typically ordered. They don't need to be, but you can order them just for convenience, from high variance to low variance. In other words, the first one has the highest variance, meaning that if this turns out to be noise, for example, and then you remove it, you get most of the noise. Okay, that's kind of how much variance is explained by each one of these components in the signal, basically. And since each one of them is a combination of these channels, you can also plot a heat map corresponding to each one of these components. For example, the first component is the heat map of the first component is plotted here, and you can see that it's mostly the frontal cortex. Okay, and it basically gives you an idea about the distribution of this signal, the component, the ICA component, which is mixture of signals, across the head map or the skull, basically. For instance, some of these, it's a very informative to look at these skull maps, because that can actually tell you where the component is concentrated, and by just visually inspecting these skull maps, you can identify which one of these components need to be removed, because they might be due to eye blinks, they might be due to muscles, they might be due to an electrode, having a poor connection with the skull, and therefore, after removing some of these components, you can apply demixing. In other words, you multiply with A again, because these you believe to be the source, right? Now it's a forward problem, but you already estimated A, so you can use A to demix it to generate the deconstructed EEG signals, which is shown on the far right. Okay, it's a good practice to include the I channels. For example, here EOG1, EOG2 represent the I channels, but not the references maybe. But good idea to include I channels, because I channels have artifacts in them, and these artifacts should appear as one or more ICA components. So therefore, you can identify them easily and then remove them. Okay, so I channels, if you include the I channels, it also, like if there's a channel, for example, correlated with the I channel, that channel's part of that channel is going to be the same component. So if you remove the component that is due to eye movements, that will also remove the artifact that corresponds to, for example, let's say O1, that shows a correlation with the eye channel. Okay, so this, for example, negative peak can be also seen, for example, at F7. But if you remove that component, that will actually take away this artifact as well as this artifact together. Because as I said before, these components basically are mixtures of these channels. Removing one of them with their corresponding weights can remove artifacts from each one of these channels. Okay? So ICA can save you from some subset of artifacts, not all of them, particularly the artifacts that are easy to identify, such as eye movements are one of them. Muscles are another. But there are also other artifacts, like it could be due to your mimic, or it could be due to your brain, for example, not focusing or paying enough attention to the task. These also create different artifacts in the EEG signal. So therefore, you have to use artifact removal algorithms, which I'm going to emphasize or touch upon one of them today. But just know that there are multiple other artifact removal techniques. I think we have to be really careful about using artifact removal techniques, depending on which field you're working. People approach the artifact removal differently. For example, computer science people mostly are fond of imputation of data, which means you generate new data based on the available. After removal of some of the artifacts, they generate new data based on the available. However, psychology department people, for instance, they are reluctant to do the imputation. They're more likely to remove the data and then use the remaining available data set. So it depends on what you want to achieve, and also the which field you're working on, because people pay attention to different things when you present your data to them. So here's an example that how I did the removal of two different IC, the independent components, and the rest of the two are demixed to generate the corrected or artifact corrected EEG. Hopefully, you will have a better and more stable EEG after this removal operation. ICA is probably the standard technique right now with respect to pre-processing EEG signals. It's not something uncommon. So I believe that in any of the tasks you do, whether it be simple ERP, which I'll talk about today, or could be other paradigms, like instead of an oddball paradigm, you may employ maybe visually evoked potential paradigms, and like flickering the stimulation. And in any event, you will have to use ICA to identify these obvious artifacts and remove them from your signal. So here are some examples, again, like due to eye blink, muscle activity, impedance, or neural signal. Like the neural signal is the brain signal that we would like to focus on. But heartbeats, for example, horizontal eye movements, these are all obvious artifacts that need to be removed.
So that neural signal is not an artifact.
No, it's not an artifact. It's just another component that I just wanted to show here. That's what we would like to focus on. Yeah. The IC label I mentioned earlier names it differently. It says it's brain, so it's brain signal, which means the original brain signal. So based on the framework you used, the naming convention that I used here might be different, just so you know. And again, through experience, you can identify these problematic cases by just looking at the Scott map. Based on the Scott map, for example, look at the impedance case. You clearly see that some electrodes having issues with their contact impedance, or maybe the conductivity. The conductivity can manifest itself in one of these components. For example, we're using in our experiment 64 channels. That means because of the full rank constraints of the demixing matrix, you will have 64 ICA components. And I will ensure you that if you've got a problem with your one or more electrodes, then you will realize that some components, maybe in low variance regime, some of these EEG components will manifest themselves as the one that I'm showing you with the impedance cell. So you should be able to remove them, because these are the problematic electrodes. Okay, as I shown you, this is another example to show you the bad electrodes. You can see that they're kind of concentrated in a specific one or more electrodes. And you can also take a look at their activity power spectrum, which I'm going to talk about maybe later. But based on these spectrum, illustrations, how the signal is spread across different frequencies, and based on the Scott map, you should be able to decide, which signals, what part of the signal is due to the bad electrodes. You can also alternatively have bad channels too. It's not just so bad channels and bad electrodes are basically the same thing. But bad channel doesn't necessarily mean bad electrode. Sometimes bad channels could be due to other neighboring channels that have got so much artifact in them. So therefore, it's just not easy or maybe impossible for you to remove the artifact in that bad channel. Although it can have some information in it, but it is so embedded inside these artifacts or noise. So you better just remove it, because in the later stages of your post-processing, that can be harmful for better inference. Okay, so bad channel phenomena is like more a superset of bad electrode case. Okay, here's another map. This is like three-dimensional Scott map, basically showing you which electrode might be having an issue. Although for example, in our experiments, we look at the real-time impedance of these electrodes when we hook up them on the subject, and we are able to tell which electrode is good, which electrode is bad. But again, depending on the circumstances, like having no acoustic chamber or having no control over the subject or the electrodes, you may fall into one of these cases, and you can identify these bad electrodes using ICA. Anyway, I want to also touch upon, maybe not go into the details, but as I said before, ICA is not able to remove all the artifacts in the artifact space. It can identify some of the obvious ones, but not every one of them, particularly background task processing in the brain. So therefore, you need to use some artifact removal algorithms. And one, maybe the most popular one, is the artifact subspace reconstruction. It can either remove it or it can reconstruct it, like imputation, data imputation. The idea is very simple. Basically, it basically calculates principal component, and now it just runs PCA on a window that's a moving window, but it chooses a reference window. So therefore, it basically chooses this reference window because it believes it to be clean and represents the clean portions of data to serve as the reference. Then you apply a principal component analysis to this moving window to remove artifacts relative to the selected reference. Okay, so the intuition behind this approach is that meaningful brain signals across channels, especially those clustered in a particular scope region, should have low variation relative to these artifacts, so that artifacts are detectable relative to the regions with minimized variance. So there are some prerequisites to run the artifact removal algorithms, particularly the subspace reconstruction method. It requires data to be already low and high pass filtered. For example, signal cannot have non-zero mean. Data is assumed to be full rank, so cannot use common. If you use, for example, common average referencing, again, this is one of the previous steps that we run. We typically have mastoids to use reference signal, but if you use common average reference, then that can cause correlation between the channels. Therefore, your data may not be full rank, and therefore the outcome of the ASR may not be reliable. So it's best to remove the mixed channels, meaning that these mixed channels usually used for trigger channels or packet counter or flatline channels, for example. Some of the channels that if you haven't removed them, I will remove them in my example with the EEG lab. But if you don't remove them, for example, flat channels, for example, could be one of your mixed channels. So it's best to remove them before you run ASR. Keeping EXG channels, such as I channels, for example, should work, but it is better to test with and without them, okay? So it's kind of, there is no standard here. It's advised that you both use the ASR, both use for with and without the presence of the I channels, for example, or for instance, the reference channels, if you have any. And finally, the final step in the pre-processing is called epoching. The epoching is nothing but segmenting the data. So you have to make sure that you do not lose any important information, and the baseline segment is still long enough after cutting off the affected portions of the signal. In other words, you have to basically put a window between the tasks that you're running, such that you can use maybe some or maybe all of these non-relevant windows of data to do the baseline correction later on. The baseline correction is needed because when we basically remove the DC component in the EEG signal, such as we run low pass filtering, right? It basically removes the mean or reduces the mean to zero of all the entire spectrum, time spectrum of the EEG signal. But if you do the epoching, which means you basically cut the signal into windows, simple segments, then some of the segments may have non-zero value, right? So you don't know which the axis of these signals, like should it be zero, should it be something close to zero, or maybe way larger than zero? To be able to understand that, you can use these non-relevant windows in the time spectrum of the signal, and then you do baseline correction, meaning that you calculate the mean on these non-relevant windows and then subtract that from the relevant portion of the signal. Okay, so the segments should not be too small. For example, up to 1 second maybe could be a good practical value, but it should not be too long nevertheless, because the longer the signal, the worse the artifacts, right, could become. Particularly if you think about in a phase coherence perspective, unfortunately, EEG signals, as I shall talk about later, it can be decomposed into different frequency components, and different frequency components can have different phase based on the stimulation. That's why we have, for example, brain signal bands like alpha band, beta band, different bands correspond to different frequency components in the signal. And they are very useful, for identification of neurophysiological, phenomena or the cognitive processes that the brain is running. Like, for example, in sleep, when you sleep, for instance, it basically changes the EEG frequency spectrum dramatically. However, if you go beyond 1 second, then the phase difference between these frequency components is so large that when you add them together, then the signal is no longer reliable. So therefore, that is due to this phase difference of not just the brain signal, but also the external signals that are contaminating your original signal. It could be due to eye, or it could be due to other background job the brain is running. They are also become desync or dephased with respect to the original signal, the source signal you're trying to estimate. And that would, unfortunately, it would make your signal essentially useless because of the phase differences. Because if you have a phase difference and you sum the signal, it will basically give you, nothing. You can't observe any information content. So here's one example that again goes back to one of the experiments we run here. We run an experiment that the stimuli consisted of clear face images, blur face images, and clear and blur object images. So we did epoch it. In other words, we segmented the data for 1 second, but we also included 300 milliseconds baseline. And as you can see that these labels and the demarcation between these epochs are shown, to characterize or localize where exactly the onset of the stimulation, and 300 milliseconds before that is used, which is irrelevant, but it is used for baseline correction, and anything after the onset of the stimulation is the signal, the original or the pre-processed signal that you would like to analyze, okay, which leads to the post-processing steps. Maybe I will skip this. This is just the background on convolution analyzing. Lastly, I'm going to talk a little bit about the post-process, one specific post-processing called ERP, because that's what I'm going to do with the EEG. I have extra half an hour, I assume, right? How much time I have? Do I have?
Yeah, you can go for the next half an hour. That should be fine.
Okay. So I'll be quick here, and then I'll use the last half an hour for the MATLAB. Okay.
Oh, okay.
So the pre-processed EEG. So we're done with pre-processing. The pre-processed EEG can be post-processed in different ways. Again, depending on your objectives. But the ERP analysis evoke response potential or event-related potential. Sometimes using event-related potentials. They're based on the assumption that there is an event as part of your stimulation set. And therefore, these ERP signals emerge on the EEG signal. And therefore, running an ERP analysis, you can just take a look at its time pattern that would come out from EEG through averaging, okay? The ERP analysis is basically averaging, but you have to be careful about, while you're doing averaging, you have to be careful about some of the things that I'll mention in my next few slides. But ERP analysis basically is a time-based analysis. It can also create a spatial information based on the scope distribution of ERP based on the scope maps, right? So for each channel, for example, you can run your ERP analysis independently, and you will have an understanding of how the ERP looks like based on different channels, which means, some sort of a spatial, understanding of the variation of the ERP. So that's why I said it can provide some maybe weak understanding of spatial resolution. The time frequency analysis, which I probably won't be able to do today, provides the time resolution as well as frequency resolution, because it analyzes the signal using two different axes. One is time and the other is frequency. Classification analysis, I won't be able to go into details today, but it gives you an idea about what's happening in time as well as in spatial domain because of the scope map. But also it allows you to understand the embedding space, because you transform your multivariate signal into another space, which doesn't necessarily need to be time dependent, okay? Like, for example, using windowing techniques. But the idea is that based on the tasks or maybe conditions within the task defined prior to experiment, based on these conditions, you can actually do a little bit of an averaging, for example, using ERP. Then you create your data vectors or data arrays or vectors, and therefore you run classification based on these data arrays to understand, how separable these signals are. And that creates, in the embedding space, since you do a feature extraction based on this data arrays, which are purely time-dependent samples, would allow you to understand in the embedding space how can you make these two different conditions separable in the embedding space. And finally, there are various statistical analysis tools such as source analysis or connectivity analysis, which I won't be able to again go and get into detail. But they actually provide you time, frequency, and embedding space. So there are a lot of fancy ways, and if you look at the maybe most recent publications, you will find lots of fancy ways of doing post-processing. Like each fancy way basically turn into a publication. But they have got like specific purposes for each one of these approaches, and they seem to defend their standpoint. Based on the previous techniques, for example, not necessarily addressing that specific problem, and therefore we come up with a new technique to address that specific problem. So it's an ongoing research, okay? So that's why I had the hardest time to classify what type of post-processing techniques available in the literature, and I come up with these four. And for example, statistical analysis is a large universe of techniques that it's probably not going to be able to capture by just saying that there are subclasses like source analysis, connectivity analysis. Like, for example, causal analysis is yet another one, and it's a very difficult one because understanding the difference between the correlation and causality is particularly a challenging one, and it's still ongoing. There are lots of ongoing work on that branch. Okay, quickly, event-related potential is EEGs or ERPs cannot, like they can reliably help you understand the neural activity across time, the time axis, and also help you understand the sequence of mental operations. Unfortunately, sequence of mental operations, it doesn't necessarily mean they're causally related. As I said before, you may need to, for example, run some connectivity analysis first to be able to understand the causal relationships between these mental operations. Unfortunately, ERP cannot precisely tell you what exactly in the brain the neural activity is originating. As I said before, even you run ICA, it's not reliable, and so therefore there are other techniques to locate the exact brain location, to locate what the brain stem location where these signals are originating from. And EEG cannot, or ERP cannot tell you, for example, the frequency spectrum of the signals either. Anyway, so there's some history here. Let me just jump. Okay, so one specific feature of the ERP is that they have to be event locked, or maybe I can tell you, I can actually decompose this. Event locked meaning that it should be time locked and also phase locked. Okay, so time locked meaning that the time between the onset of your stimulation and the time the response starts showing some effect should be the same, okay? Like it could be either across trials or it could be across different subjects. If the time difference occurs, for example, averaging across subjects is not something recommended. Why? Because some subjects may show delayed response based on their, for example, if they've got they might have different impairment, like if you compare, for example, a impaired brain and with a neurotypical, you will realize that some of these impairments could cause delay in the response. So therefore, they're not going to be necessarily time locked or phase locked too. Like the phases of the different frequency components of the EEG signal can show differences between different brains, okay, depending on their health. Okay, like even if you are psychologically under pressure, like for example, before exams, we used to be when we were students. If you look at the EEG, for example, under pressure, under psychologically pressured brain, you will realize that this phase lock assumption is no longer true. Unfortunately, the signals start showing phase differences in different frequency domains because of could be a threat or could be some other external phenomena that's modulating your EEG. So when you collect your EEG signal, you have to make sure that the subject doesn't think about anything else or are not under, for example, influence of caffeine or other important stuff. That should make the idea behind all these requirements is to make sure that when you run an ERP, you have a time lock and phase lock signal. Okay. Okay, maybe I should stop here because there are many more things maybe talk about the ERP, but I don't think they are necessarily relevant to my example, MATLAB example today. So maybe I should stop here, but I may have to go continue maybe later on. So let us just talk about how do we process signals using EEG Lab. Okay, so let me see if I have a source signal already. Maybe I should have also shared some example source signals with everyone. Yeah, let me share them if I have them handy. Let me see where they are. Let's see. Yeah, that's my bad. Sorry about that. Let me see. Yeah, so this is SSVEP though, but that's still okay, I guess. How can I share this one? Where is it, MATLAB? Okay. Should I share pretty right now with you? Would that be all right?
Yes, then I can just circulate to everybody.
Okay. Whatever you want. I mean, I can share it now or I can share it after the presentation just to save time.
After is fine too. I mean, that's fine. I think people would become more
Okay.
comfortable doing it by
Let's do it the following way. I'm going to, so this is recorded. So I'm going to show you some pre-processing steps together. We'll do it together. And then you watch the video and you try it yourself with the input data that I'll share after the meeting. And if you come across something unfamiliar or something that you don't understand, or maybe shows different results, then we can discuss it in our next meeting. Would that be all right?
Yeah, sure.
Okay. All right. So now I'm going to show you one specific example. So we have a biosemi setup in the downstairs here in our department, and so therefore our output EEG output signal is formatted in the BDF file format. So here you see there's a BDF, but you could have other formats, just so you know, depending on your setup and your capture device or amplifier. I think it comes like together, but depending on your brand, they may adopt different data formats. Okay, so BDF is the data format the biosemi use. So I'm going to use that one. So if you just click on it, you will actually open this window to tell you where the signal is or where the data is. So here is one of those the data set that I generated before, which is based on the flickering stimuli. That's why you see SSVEP, which means steady state visually evoked potential. That's not recommended for everyone because if you've got an epilepsy, it can lead to some unfavorable circumstances or outcomes. So if you just click on the data set, if you choose the data set, then you'll be asked about some details about the data set, like extract the event from marker channel, force continuous data when data is stored in blocks, and etc. I would recommend you do not touch these default selections for now, okay? So don't worry about this window, because you can fix every single one of them later on, okay? As we understand more about how to use the software, as well as get some more familiarity with the data set, then we can go back and change every one of them, okay? So I would just recommend just click on OK for now, but then we will worry about them later. Okay, now I'm just waiting because it's reading the data in BDF, okay? Okay, one thing that I'd like to mention to you that some data sets, they come with the event names or events or the triggers, they're already embedded in the data set. Sometimes it comes externally. So depending on your setup again, you may want to import your events or labels or events locations from an external data, maybe an external data text file could be a potential format for that. But it again depends on your setup and in your on the inner workings of your or your device. So in our case, the event names, their precise locations in time, they all come with the data. So they're basically part of the BDF file. Okay, so that's why it says importing data events and etc. So here I'm going to name my data. So I'm going to just say test1, okay, and then click on OK. So I already loaded my data, and you can see that test1 is right here, and it gives you some information about the data set. For example, there are 72 channels, frames per epoch, which means you have, since we have an epoch, there are so many of them because each one of the samples is considered as like one simple epoch. So that's why you have the epochs is just one, and these are corresponding to the number of samples you have. And the 44 events, because these are the events that we introduced into the BDF file. And maybe I'll talk about them more later on. This is the sampling frequency. So you have 2,048 samples per second to represent your signal. And reference is unknown because we haven't re-referenced it. No channel locations, no ICAs run, and the data set size is 306.2 megabytes. Okay, and as we do the processing, as you shall see later, the data size will drop. Okay. All right, so I'm going to show you this, maybe remind you, because I already have shown this before, but I'm going to remind you that we had this pre-processing. This is not the one. Sorry about this one. This one. Yeah, no, it's not it. Maybe this one. No, there is my. Oh, this one. Okay. So you may remember maybe earlier this pre-processing steps. This one. Okay, maybe I can make it bigger. So this pre-processing step is just not standard, but almost close to the standard. And I'm going to tell you where to be more cautious of taking these steps. So I already imported the data, and then I come here, I examine to leave out the data, the EXGs. I told you that about like for ASR, this may be a requirement. And we would like removal of redundant channel information, but not necessarily artifacts. So let's just do them together. I think I've got like 20 more minutes. So first thing we do, basically you just take a look at your signal, okay? So you basically go to plot, and then you plot your signal, and you see nothing, right? I mean, something, but not necessarily they're all aligned in the same axis, right? So therefore, you go to display, you remove the DC offset, okay? And then now you will be able to see your signal, like all 64 in line. One thing that you realize that we have extra EXG channels here, or MISC channels could also be named as MISC, but 5, 6, 7, 8. We need to remove them because they have got no information in it, not even nodes, because we haven't used those electrodes. Okay, and that can also happen to you. So we're going to remove them. And then the removal is pretty easy. You go to tools, you go to edit, and then you go to select data. Okay, now if you want to remove, you have to click on here because it says on remove these, right? And then you click on here to choose which channels to remove, like ext5, and then I press on shift button to choose multiple. So I choose ext5, 6, 7, and 8. And then click on OK, and then you click another OK. Okay, then it will ask you to rename it. Most likely, since you don't want to go back, right, you can just override it and use the same name. Okay, that will save a lot of space on your memory. So you can actually click OK. Okay, and then if you would like to see your signal again, excuse me. Okay, and then you have your signal now displayed, and if you remove the DC component, you will realize that there are no more extraneous 4 channels that are at the end of this channel list, right? And one thing that you might already realize that these channels are already named, right? You see FP1, AF3, and etc. In many cases, they're not named automatically, so you may need to name them using channel location information. You can actually go to tools and tools, and then you can actually use where was it? Well, maybe here, channel locations, by name. Okay, so you can actually name these channel locations by name or by number. Sometimes the automatic naming use A1 and B1, like A1 to A32, B1 to B32. But you can change those names based on your location file. Particularly, the location file is very important because it will tell you where exactly these electrodes are physically located on the skull. Because if you want to generate realistic heat maps on the skull, then you need to give this mapping information with a certain level of accuracy. Otherwise, your scalp map you generate wouldn't map correctly to what's actually happening on the brain. Anyway, so let me skip that one and then let's do the re-referencing, okay? So here is the re-referencing, and I'm going to re-reference my signal using re-reference the data from the tools. If you click on it, you've got another window, and then so there are 2 options here, as you can see. You can compute the average for re-referencing, but if you don't have mastoids or electrodes that are on the mastoids, I will recommend you do this one because there is no other choice. But rather than that, I'm going to re-reference it using the mastoids because I have those channels, and it's right there here, M1 and M2. Okay. Oh, sorry about that. I haven't chosen yet. Yeah, let's just say okay. All right. And then, so there are options like interpolate, remove, because M1 and M2 will be removed. Do you want to interpolate them? Well, I don't want. I don't need those extra channels. Or you can retain those channels as part of your processing. But most of the time, mastoids are close to the skin, and therefore the brain signals or information content of these M1 and M2 channels are pretty poor. So therefore, it's recommended that you remove them. Okay. So less number of channels means more precision in the later processing steps of the pre-processing phase. Okay, you click on OK. I don't know what's happening. Oh, sorry about that. Yeah, so I think I've done something wrong, so I need to do it again. think I just clicked on OK twice. That's why it didn't go through. Okay, now it should work. All right, so if you, so again, it asks you to, have another copy, or you want to overwrite it. I'm going to overwrite it. So overwriting means if you click on the data set, you will see that there's only one copy, okay? Because I've overwritten every one of them. But you don't have to do this, and as I'll show you, as you go process step by step in the pre-processing pipeline, you will realize that some steps you may want to store the data as two different copies, so you can have different branches in your pre-processing. Particularly if you change the experimental paradigm, for example, rather than showing clear faces or blue faces and etc. If you flicker the data or flicker the stimuli, like an image, for instance, then it's a good idea to not follow a standard pipeline, but rather try other possible potential alternatives to make sure that your pre-processing helps your post-processing. This is kind of advanced, but just so you know that it's a good idea, sometimes good idea to have different copies of different branches of the pre-processing pipeline. And this data set option here provides you that flexibility. Okay, so next is the filtering. So I'm going to just do the filtering. So you go to tools, you filter the data. You select basic FIR filter for now. Okay, and it tells you the lower edge and higher edge of frequency passbands, so how much frequency you want, how much frequency you want to just take out or remove from the original signal. I'm going to just put 0.5 hertz as the lower edge of my frequency. So that means anything below 0.5 hertz will be removed from the signal. Okay, and that explains why we have these extra DC components. That's why we're not able to see EEG signals on the same axis. So we can basically compare across different channels because they're basically everywhere due to different DC components. But after removing the low pass content, that means we could remove the DC component and we will be able to see better visualization of the EEG signal. So don't worry about the rest of the others. So just click on OK. And this shows you the frequency response of the filter. But don't worry about that. So I'm going to just, because this requires more explanation, I don't want to spend time now with the explanation. But you can see if you look at your command window on the MATLAB, you will realize the progress of the low pass filtering that's applied to the EEG signal. Unfortunately, my computer is a bit slow. That's why it takes so much time. But if you have got any questions so far, I'll be happy to answer. Okay. If you don't, maybe I can just talk a little bit about the convolution operation, which is here, that I skipped before. So here's the convolution operation. Convolution is basically very essential, not just from engineering perspective, but from vision too, because it is kind of inherent to our visual system to run convolution operations through ganglion cells, for instance. So convolution is a basic functional operation that a discrete time, linear time invariant systems employs to create its output. Why am I talking about convolution? Because we have these LTI systems in our pre-processing, such as filtering. The filtering is an LTI system, basically. It has got an impulse response, and you convolve this impulse response of the filter with your input signal to be able to filter the frequency components of the inputs. So this particular sum is fairly easy to describe on the right. You can see that the input is very simple Dirac function, like a single one that's concentrated on a specific index I. That goes through the system's impulse response, which is characterized by the H function, what you do basically, you take this impulse response function of the filter, flip it, okay, and then move it as like a moving window over the input to calculate the output. Okay, that's what this equation tells you, or both in sum or in the integral form. But in both cases, this type of time domain operation results in the frequency domain as a filtering operation. Filtering, it doesn't need to be low or high pass, which means you allow the low components to pass or high components to pass. But it also can allow you to some bands, for example, to be stopped or passed, okay, depending on your impulse response. This is a discrete time, discrete domain example, but and you can have continuous because it's simple to illustrate with a discrete domain. That's why I've chosen this example. But you can think of this as a continuous waveform too, like the input and output. They're both continuous waveforms, and you can flip the impulse response of the system, which is continuous, and then basically move it over the input to calculate, which corresponds to the integral operation. So convolution is this basic operation that does the filtering operation effectively. So it can be combined, okay, actually defined in both in discrete and continuous time, as I said before. But it's the basic operation behind the filters. That's why I wanted to mention it. And let me just skip to this one. Yeah, there's this very interesting phenomenon called aliasing. I don't know what the status of my filtering. It's almost there. So I'll talk about aliasing in a minute. So when we do maybe run ICA. So let me just go back to my pre-processing. So do you want to override it, or do you want to save a separate copy? Well, I'm going to override it because filtering is so basic that it's going to be probably part of every pipeline you can conceive. So therefore, I'm going to just say OK and override it. Okay, so now I have the filtered version of my signal. You go to plot, you just click on channel data visualization, and boom, you don't need to now do any DC adjustments because your data is already filtered, which means you basically stopped the low pass components of the signal and you basically let the high pass pass. And therefore, you have the signal now all aligned the same axis. And therefore, you can visualize your EEG signal. Although it looks a little complicated because there might be some artifacts going on, but you can move to the right to see the conditions. For example, you can see there are these conditions, which basically corresponds to labels, these events, and each need to be epochs at the very end of the pre-processing step. So let me just move on quickly to ICA just to finish quickly. So let's just go back to this over here. Okay. So one of the things that you may want to worry about is cleaning the line frequency, which you can actually take a look at the frequency components very easily through EEGLab. So let me show that too quickly. So if you look at the plot and then you go to channel spectra and maps, okay, that will allow you to see the spectrum, the frequency spectrum, which I haven't talked about. But frequency spectrum of a signal can be found using a technique called Fourier transform, which I'll probably go through if you're interested maybe later on. But with basically it tells you what components of frequency are available in your signal, in your time domain signal. I'm finishing last few minutes.
Because I left Pretty as host because I'm going to have to do.
Okay, got it. Okay. So here you may want to change here some of the selections, like this basically tells you what's the spectrum you're interested in. So I'm going to basically say 0.5 to 80 hertz. And if you want to, so this is the range epoch time. So yeah, let's not worry about the rest. So it's going to basically plot you the frequency content of the signal, and this could be really useful because it would tell you what component, and particularly for clean cleaning the line noise, right? The line noise typically is at 60 hertz for US and 50 hertz for Europe. And if you don't have like acoustically isolated chambers, you will be able to see this kind of line noise. It's pretty standard to see this. Okay, here is the plot. It basically shows that for every single channel here is transformed in frequency domain. And you clearly see that there is no line noise here. Like if you had the line noise, you would see the peak here, and therefore needs to be removed using something called notch filtering. But here, as I said before, this data is collected in our downstairs where we have this chamber. Therefore, we didn't have any 60 hertz or 50 hertz peak. But if that's the case, I recommend that you should remove that. Okay, just so you know. And then yeah. So I'm not going to do the cleaning the line noise. I'm going to just skip it. So the other thing is that you want to remove the artifacts before maybe the ICA. So I'm going to skip this section for the reasons that I explain maybe later. But this helps you identify other types of artifacts that the ICA wouldn't be able to find. And for example, background brain operations. Or it can identify bad channels, okay? And that helps you identify them, but not only identify them, but also remove them. But including, you can also identify I channels too, or I artifacts, but these are already can be taken care of by the ICA later on. So I'm going to skip this ASR because ASR can be applied before ICA or after ICA. So both is okay. Although your approach could be different in those two cases. Anyway, then just keep the story short. I'm going to just skip the ASR part, but then I have to do the downsampling, right? Downsampling means you basically decrease the number of samples in your data set. So you can see that your data set is around 281.4 megabytes. As you execute each one of these steps or blocks in the pre-processing pipeline, your data set basically gets shrinking and shrinking more. And downsampling is one of that major step in the pre-processing that will shrink your data set even more. So what you do is basically you go to filter, you go to the resampling. So you see change sampling rate. And it's 2048 samples per second. So you want to change it to maybe 512. Okay, I'll tell you how you do this, but 512 is more than sufficient for many of the EEG signals. It's high enough to retain the maximum frequency of 256, but most of the time, anything beyond 256 hertz is not very useful from an EEG perspective. Some people use 256. You can also use 256, but that means the maximum frequency available in your data must be lower than 128, which is the half of this. Again, due to something called Nyquist frequency and aliasing discussion that I was trying to allude to before the filtering operation. But we can talk about that. That's due to this aliasing phenomena. In order to avoid these aliasing phenomena, you need to choose your sampling rate such that the maximum available frequency, which is useful to you or to your processing, half the sampling rate. Okay, so 256 probably is also okay because I would not expect to have information to be contained above 128 hertz. Okay, it most likely my information would be residing in between 0 to maybe 80 hertz or maybe 70 hertz. Okay. Okay, so I'm going to just make it 256 so that my ICA time will be less because ICA is based on the number of samples that it processes. So the lower the sampling rate, higher the ICA efficiency. So I'm not going to overwrite this because it's a resampled version. Maybe I want to keep the original. So I'm going to just click on OK. And yes, here we go. You can see the data set size is maybe reduced by factor of 10 now, right? It's just 35.3 megabytes now. So basically get rid of lots of data, but they are most likely not very useful, okay? And then next is the ICA, right? So now I have to run the ICA. So there are multiple options here. There are multiple implementations of ICA, and one of which is called, it's very well known, but it's called the ICA MA, but I probably will not run it today because it takes more time. But in order to run ICA, you choose decompose data by ICA, and this version of run ICA is the default and maybe the most efficient one. There's this faster version and also slower version, so whichever you want. But there are also other options you can see here, which is based on this Suayb and Jade and executable version of the run ICA. And I see this the other version that I just mentioned is a plugin. So you have to set that up if you want to run that one. And I'm going to show that one as well. So let me just show that one. So it's right here. It's called Amica, ICAMA, or sometimes called Amica. So the this is available through the plugin section of the EEG lab. You can download it and you can set it up and then you can run it, but you will see that it takes more time. And that's why I'm going to just use the default version for now. So it's pretty simple. You basically run ICA, you choose this one, and then you don't touch anything else. If you want to run this ICA for different channels, then you can choose channels actually. Okay. Here I already removed most of the irrelevant channels, and I want to include the left eye and right eye channels. You can see LE and RE. Because I want it to be part of the ICA that can help me identify accuracy, increase my accuracy in the identification of the different components. So I'm going to keep them and then just click on OK. So you can see that the ICA takes time. It's going to run a lot of iterations because as I said before, calculation of mixing matrix and the source is a challenging problem. You have to make assumptions, but those assumptions result in an implementation that does lots of approximations. And therefore, most of the ICA implementations are done in an iterative way. And an iteration is subject to, of course, a certain convergence criterion. And you can see that from the run, there's a learning rate that you can change. And also that learning rate causes some changes in the convergence. And therefore, you will just look for this change becoming very small. If that change becomes very small, like close to 0, then it will converge. And therefore, the ICA algorithm, this run ICA, will end its processing. It takes about 300 to 400 steps. So therefore, it would take maybe a couple of more minutes. So that's why I'm going to just go back quickly to my discussion about aliasing, maybe, and then I'll finalize my talk today with the ICA outputs. Okay. So let me just jump to here. Yes, this one, this aliasing. So aliasing is basically let me tell you this. So if you're given a signal, even at discrete time or continuous time, doesn't matter. You want to sample your signal. Okay, that's like, for example, downsampling is one resampling technique. Downsampling means rather than taking all the samples, you basically take one sample at a time and then give a break. Okay. For example, let's take a look at these sinusoidal signals. The one on the right bottom, okay, it's a very highly densely sampled signal. Okay, you can see that the sampling period is very small here. And then those data points you see are basically, they're supposed to represent the signal, okay? But if you basically increase that sampling period, then you will see something like shown on the left, okay? And then the one on the left, and then the left here, okay? Until you get here. So the question is, what must be the number of samples that should I retain to be able to reconstruct the signal again? Okay, that's called the sampling theorem. What happens when you downsample and resample the signal in the frequency domain? It basically corresponds to, so every discrete signal has a periodic frequency spectrum. Okay, for example, here you see some frequency representation of a discrete signal. If as I said before, just take my word as granted. Okay, I don't have time to prove my claim here, but just believe what I say, that every discrete signal has a periodic frequency spectrum, which corresponds to something like this. And if you do downsampling, what happens is that you expand the frequency representation of these signals in every one of these axes. You can see there are replicas of it, and each one of them starts expanding. Okay, so what happens is that if you expand them enough, then they will start overlapping with each other. The question is, you want to downsample it. Like if you increase the downsampling ratio, which means the period increasing, that means you have less samples to represent your signal, but that causes your spectrum to expand. So your objective is to expand your frequency spectrum of the signal such that it doesn't cause any overlap with the other replicas. Okay, because if you have an overlap, that causes something called aliasing, and that degrades the signal. So you have got the option of to retain a number of samples, like less number of samples, until you hit in the frequency spectrum, you hit the other replica. Okay, so that's the best you can do if you want to reconstruct the signal again. So you'd reduce the number of samples in your signal until you hit the other replica in the frequency domain. So that means you stop. Okay, that means that's the best you can do. That's the smallest number of samples you need to be able to reconstruct the signal. Okay, the reason that I used frequency domain representation of the signal, because it's easier to understand. But if I want to give you an example from real life about the aliasing phenomena, you can think of a car, for example, the wheel of the car, okay, that has got this little pattern, right? So if it basically moves at a certain speed, maybe faster than the sampling rate of your visual system, then the wheel would now start looking like the wheel actually turning in the other direction, right? This is due to the aliasing phenomena, because you're not sampling fast enough with the speed of the wheel turning, right? So therefore, your sampling rate is less than the frequency content of the image of the visual, and therefore you basically end up with aliasing phenomena. It looks like an illusion, but it's not. It's due to these downsampling operation that your visual system is doing. Okay, so aliasing is something that is very intrinsic to the real life, and although it looks mathematical in this slide, but it is very real, and it's very part of our everyday life. So that's why when I choose my downsampling rate to be 256, I have to make sure that the maximum frequency available in my signal is no bigger than 128. If it's bigger than 128, then I'm going to have aliasing due to this replica, okay, phenomena. All right, I hope that we're done. Not done, but we're almost there. If you have any questions, I'll be happy to answer. I would expect this to be running maybe up to 300 something steps. That's kind of the typical number I have through my experience. So we're almost there. But if in the meantime, if you have any questions, of course, I'll be happy to answer. Okay, 42. I know that I'm late. But I want to also talk a little bit about maybe the steps that I will not be able to show today, but I'm going to guide you to what to do. Let me see. Where is my? Oh, it's this one. Yeah. So after ICA, I'm going to, of course, reject some of these components. I'm going to leave it to you as a homework, and then we will do the epoching, and then that will be the end of it. Okay. So there are actually 1 or 2 more steps to go through after the ICA. But I think I believe that you should be able to do that. And I'm going to show it quickly because they're not very long to describe or explain. Okay, I think we're almost there. So you can see that it says do not interrupt, or press button to interrupt, or otherwise you should wait until this window disappears. Yes, very close. As you can see that there's a threshold, and this change here is close to 0. Actually, this was the threshold I'm using. And therefore, if it's less than that, it stops. So now we're done. So let me quickly show you the decomposition result. So you go to reject data using ICA and reject components by map. Okay. And then you click on it. Oh, okay. One step that it requires channel location information. So you basically go to, because it doesn't, so the algorithm or this software doesn't know where the electrodes are located in the skull. So in order to generate the heat maps, you need to basically tell the EEG lab where your location file is, or describe it somehow. So that's what I'm going to do. I'm going to just say these channel locations, but I think there's a better one. Here is edit. You have channel locations. You click on the channel locations, and then you choose your model. You can choose either BEM or the BESA DipFit spherical. I usually use spherical model, but I think that the other one should also work fine. So I'm going to also share this location file. Now I'm realizing that I haven't shared everything I needed to. It should be somewhere here, EEG. Where is EEG? Oh, here. Yeah, so here's biosemi64 channel .log is my file. Okay, and yes. So if you just look at plot 2D, you will see your channel locations. Okay, here. Okay, you have to make sure that your channel locations are right. If it's not, then you should be able to choose other models or change your location file. Okay, now we have done that. I'm going to just click on OK, and then hopefully see the decomposed files. I'm sorry, decomposed components. Yeah. Yeah, so as I said before, I have 66 channels. Therefore, there are 66 components because of the full rank constraint. So I'm going to just show you 10 of them. Okay, then you can do the rest. So here is some of them. Okay, just the first 10, which is the most significant, in terms of variance. And you clearly see the distribution of each one of these components. But there are 2 options here. You either use your base based on your previous experience. This could be the visual cortex and etc. And therefore you can just ignore them, or you can use them. Or as I said before, the IC label. You can actually classify components using IC label. You go to this section and then label components. You can just click on the label components and then choose the default version. Okay, click on OK. Okay, it extracts. It does a little bit of processing. It does some extraction of features and etc. And then there's a certain default thresholding, meaning that what's the criteria, for instance, the component should satisfy to be able to classify it as brain component, muscle component, eye component, and etc. Okay, So I don't want to plot all of them, but I'm going to just say maybe the first 10. But feel free to plot all of them as long as your computer is able to handle that. Because if you got like more than 32, I assume that it opens up another window. So here's the IC label results. You can see that there are 10. I'm just plotting the 10 of them. But as you can see that most of the initial components are mostly classified as brain. Okay, and you see the reliability of each one of them. Like brain is 99.9%. So you want to keep it, actually. That's what it means. If you click on each one of these components, like first one right now, you actually have more information about this classification. For example, here you see 99.9, therefore muscle, eye, heart, line noise, channel noise, and all other artifacts basically get 0%. Here is how the trials look like. You see the trials on the y-axis, and the x-axis is the time progression. And this is the activity, the average activity at the IC1. This is which means the first component of the time representation of the first component. And this is the frequency component, frequency representation. You can see that there's a little bit of 60 hertz could be considered as line noise, but the power level of it's less than minus 10 dB. So which is pretty low. So you can actually ignore this. It's not a big deal. But it's usually in the visual part. As you can see, that's predominantly on the visual cortex, and therefore this can be considered as valid data, okay, or valid information. Whereas here, this one, the muscle, you will realize immediately that the signal shape is basically very noisy, right? That's the one thing. The other thing, if you look at the frequency spectrum of the signal, you can see that a lot of high frequency. You can see it's actually above minus 10. And also, you can see the correlation in these trials between, particularly some of the trials are very correlated with each other in this component. And that's why you see this red and blue, they're basically contiguous with each other. So that kind of gives you a more confidence about classifying this component as belonging to muscles. Okay, and you can also see the eye. The 10th component turns out to be the eye related, and it's kind of very obvious because you can see if you look at the time progression, it basically has these jumps, right? And jumps are typically due to eye. And you can also see that there are these heat maps showing somewhat closer, location to the eye. And also the continuous data based on trials also show similar trends. So the way you remove them, you basically go to tools. That's my last word. I'm going to quit. So if they remove that, you basically go to reject data using ICA and reject components by map. And I'm going to just worry about the first 10. Okay, and then click on OK. And it will basically plot you these 10, but with the IC labels pointers for you. Okay, since I run the ICA, IC label, that's why you have this reliability metric, which is plotted with the topo maps. These are called also topo maps, topological maps. And with topological maps, you have the information about which component belongs to, which components belong to which class of artifacts. And now you can actually click on the muscle, for instance, but now you have a different window because this window would allow you to accept this component or reject it. Okay, you just click on this accept button and it becomes reject. Okay, and then you can actually now close it. I'm sorry. You have to click on OK. My bad. Sorry about that. So you basically have to first click on accept. It makes it reject. And then you have to click on OK. Now you have this color change showing you that it's now labeled as reject. And I'm going to also reject the eye. Okay. and you basically click on the accept and click on OK. Okay, now I already labeled them, the two. Okay, you may want to worry about other components as well, but this is just to show you that you know how you do this. So you have to go to plot. I'm sorry, tools. Sorry about that, tools, and then you go to remove components from data. Okay, when you do that, it will automatically realize that the components 9 and 10 are flagged for rejection. That means, and then question comes: do you want to remove these components, and etc. Okay. And then you actually click on yes. And then I'm going to say again, accept. Okay. And then I'm not going to overwrite. Okay. I'm going to have another copy just in case. Okay. Then we have the ICA, components, two components removed. And if you go to plot the data, okay. Actually, I should have, let me show you the data. So that's the good thing, that you have a copy. If you go to this one without the ICA and plot the data, okay, let me just plot first the without the ICA so that you can see the difference. Okay, let me just expand my window for you. And then you go to stack. You see the stack here? If you stack the signal, they basically are now sharing the same axis. And then you move to the right. Okay, and then you will realize that some of the signal components is showing some digression from the, this one, for example. You see this one or these ones, okay? So these digressions are indicator of, like, for example, this one, you see? So these digressions are indicating that there's something fishy going on in the EEG signal. And therefore, the ICA should not show these weird looking signals. So if you just go back to your ICA process data and then look at it again, it will show you cleaner data. But the way to see it, maybe you should just stack it. Okay, although there are some still little flip-flops here and there. Okay, that means you have to remove more components basically. But eventually, after removing all these components, then you should be able to get rid of most of the artifacts. So I'm going to leave it to you just to play with it and remove more components, so that you'll be able to see cleaner signal. So you don't have those artifacts visible. Finally, I want to just do the following, because that's the final step. So I want to just close the whole session, pre-processing session, if you allow me 2 more minutes. So finally, you go to after the ICA, and then you're happy with the results. Then you go to extract epochs. Okay, so the epoch is pretty easy to do the extraction. You basically have to choose a specific, label. Those labels right now, not very telling. Particularly those conditions correspond to specific events in the experiment. So you need to make sure that you rename those event names. So I would like to just focus on, for example, let's suppose that condition 1 and condition 2 corresponds to the same event. Okay, so rather than processing all of them, you can basically process, a subset of them, or you can process all of them. Okay, so if you basically don't choose anything, it will process every one of them. So I'm going to do the processing for all of them, but you can choose. So now I'm going to choose the baseline, which is the 300 milliseconds before the onset of the stimuli, and after that maybe 1 second. So that's my epoch limit, start and end. Okay, and then I'm going to just click on OK. All right, and then I'm not going to overwrite. I'm going to keep the copy. Okay. So now the baseline correction. This says basically I'm going to use the 300 milliseconds before the onset. I say OK, because that's what I agree on. So this is removing the baseline. And then where am I going to save it? I'm going to just say epochs baseline. Okay, so I'm going to have another copy. Okay, click on OK. and then you're done. If you just look at the channel output, you will realize that you have epochs here. For example, this is the first epoch with this condition, whatever the condition is, and then this is the next epoch, right? And so these are all segments of data, basically extracted from your EEG, and those segments now accessible and can reliably post-process. Okay. For example, probably in the next session, if we ever have it, I can show you like how to do the ERP, okay, that I talked about today. Unfortunately, we couldn't have time to do any post-processing, but this is the end of the pre-processing. At least you know how to do the pre-processing now to get these segments of data for later processing stages. Particularly, one thing that I would like to mention, that post-processing doesn't require you to use GUI anymore, because it's all segments of data, and then you can access these data using just EEG's data structures that is generated, okay? And then you go to data here, okay? And then if you look at the size of this data, you can see that it's 66, 303, 33, 44, okay? Which means 66 number of channels, 303, 33 is the number of epochs, and there are 44 events. So for each one of these events, you have these matrix of information. 66 by 33. I'm sorry, 333 is not number of epochs, it's number of samples. Okay, so 66 by 333 is your total data for each one of these events or epochs. Okay, so now if you'd like to do averaging across trials or across channels, then you can basically use these three-dimensional data using source code, like pure code, like through interaction with the command line interface without using the GUI. Okay, and if you have any time, maybe in the future, I can show you how I did that. But you can also try it yourself. Okay, pretty.
Sure. Thank you, Shoaib, for such a detailed and interesting session.
Yeah. So if you have any questions, you can ask now, or you can ask me through email. Just feel free to reach out to me through email anytime you like. Don't worry about if it's something that you think is very trivial, but I will be happy to answer any questions. So don't worry about it.
That's very sweet. I think everyone will take up on that. And, when people run this themselves, they'll have more questions, which they can surely ask you.
Sure. Yes. You have to do it yourself to be able to come across, issues that I haven't touched upon today. And I'll be happy to. I've probably have encountered those problems earlier, so I'll be happy to help you with my experience.
That was great. Thank you for the elaborate session.
Sure.
And
Sure. Thank you for staying up late. I know
no, our pleasure, absolutely.
it
And
takes 2 hours today.
no, it was all fun. And if everyone gets interested in learning more about post-processing, maybe one day we can reconvene.
Yeah, we should. If everyone is happy and we're on the same page, then I would love to have another session, maybe next month or so, to continue with ERP, just to connect the whole circle.
Yes, yes, we should do that.
Okay.
Okay.
Yeah, I think you are the host, so you should stop recording first so that it will be recorded.
Okay.